Hey, everybody. Hope you're doing good today. We're going to take a look at Chapter 6, which is going to talk about viruses. So, we're going to look at, for example, what are viruses? We're going to look at some of the structures and the characteristics of what they would look like. We're going to look at then a little bit of the replication cycle, how it is they go about infecting, and we're going to look at just the beginning of how they start to duplicate themselves and what's happening there. We're going to continue this topic in lecture next week, where we go on to finish the cycle and go on to talk about how they cause damage to host cells, how they can cause cancer, for example. And then we will also look at a different type of virus called a bacteriophage, which is a type that infects bacteria. So more of a microbiology topic. So we're going to look at today these first three topics here. So to start out with, we want to talk about what a virus is. And real quick, let's just pull up a figure here. I should have had one right there for you. Let's just pull up a picture of a virus, and we see that, first of all, a lot of these are animations. It's tough to get real pictures of viruses, but they do have many, so let's find one here. Here's one. This is an infamous photo. This is a real viral image here, and this is, if you may already know, this is the Ebola virus. So what this really is here, I'm using this as the example, it's really a package of protein and DNA. The Ebola virus is a type that is going to package up its basic essentials, and really when it comes in contact with a host cell, this little bundle of protein and DNA here is just going to spring to life and start to hijack, for lack of a better word, the host cell. And actually, I need to clarify a little bit here. The Ebola virus is actually an RNA virus. RNA is very similar to DNA, but this is actually RNA-based as opposed to DNA-based. A little bit of a technical difference there. But anyway, basically it's a genetic code surrounded by protein. And in this case, there's actually also a little bit of lipid, a little bit of fat involved with it. But for the most part, it's just a bundle of organic nutrients, really, in a highly organized way. So by the definition of scientists, these are considered non-living entities. They don't have the qualities, quote unquote, to meet the description of a living thing. So rather, they're referred to as infectious particles. And this largely has to do with the fact that scientists have defined what a living thing is. And this is arbitrary, meaning it's based on someone's kind of opinion, more or less. And based on that arbitrary distinction, a living organism has to do certain things. So, for example, they have to be able to respond to the environment. They have to be able to grow and develop. They have to be able to obtain and utilize energy and consist of one or more cells. And a cell always has, for example, a cell membrane. It always has ribosomes, so they have to have all those things, cytoplasm. And the truth is, a virus has none of these things and does none of these things until it comes in contact with the host cell. So what's weird about them is that they don't do these on their own, but they do some of these things when they come in contact with a host cell, something that they can essentially infect. So that's where these are slightly difficult to describe exactly. I put together this little brief figure here to kind of give us a better perspective. If we start with just basic matter, things like atoms, not even going as far down as we could get into protons, neutrons, and quarks and bosons, but one of the basic fundamental structures of all matter is an atom, very fundamental building block of life. Atoms join in very simple ways to create simple molecules like water, methane is an example, CO2. And then these types of simple molecules can assemble to make really complex macromolecules. These are things like proteins, carbohydrates, lipids, membranes, cell membranes, things like that, complex macromolecules. So we have atoms joining to make molecules and then large molecules. Now, jump past here this real quick. These is essentially form large structures that we refer to as cells, which are a large, complicated arrangement of these macromolecules. But a cell has a membrane. It has the ability to respond to the environment. It can replicate. It takes on a whole life of itself. And then these form in multiple or many units to form a multicellular organism, complex life like you and I as humans. Okay, so we go from atoms all the way up to cells and then multicellular organisms. Viruses fit somewhere in between macromolecules and cells. They are a very, very, very advanced form of a macromolecule, but they're not nearly as complex as a cell. So a cell is this really, really, really complicated thing. It can make copies of itself automatically. Okay, so it has its own life that is built into the programming of the cell. Viruses don't have that. So a virus is something that when it comes in contact with a cell will actually attach to the cell and infect it, potentially anyway. And this is actually a real image here showing, this is artificially colored, but this is showing a bacterial virus attaching to a host cell and beginning to infect it by injecting its DNA across. It looks like something out of a science fiction movie, but that is actually real. And what they represent really is, what they really represent, in my estimation, is an interaction, an intermediate rather, between simple molecules and complex cells. They are not defined as living, but some people might argue that the definition of life could be changed to include viruses. Perhaps life maybe is simpler than we thought, and maybe this is an example. But that's really more of just an opinion. There is no right answer there. As it's defined today, I'll go back here real quick. This is as it's defined today. So this is the technically correct way of looking at life. And by looking at it that way, we exclude viruses. But that's kind of where they fit here in the spectrum. More than molecules, less than cells. Okay? So that part is relatively unimportant to really understanding how they work, but more of a philosophical understanding of what they are. And to be fair, it's not that easy to define exactly what they are. They're an interesting aspect of life when you really look at them technically. Now, another interesting thing about viruses is that we typically think of them as agents of human disease, as that first bullet point indicates. But the truth is, viruses infect every living organism and cell type on Earth. I say every type. The vast majority, are subject to some kind of viral infection. Now, the truth is, viruses come in lots and lots of different shapes, forms, sizes, and specificities or preferences for different host cells. So in other words, not every organism is susceptible to the same viruses. We have some viruses, for example, that infect humans, some viruses that only infect cats. Then there are some viruses that only infect plants. And then there are other viruses like this one down here in the corner, which is another real image, that only infect bacteria. So while every cell type is susceptible, each cell has its own susceptibility in terms of what type of virus will potentially infect it. That's what this statement here is indicating. Okay, but really there's no exceptions. Viruses, there actually are even examples of viruses infecting other viruses. And that part we won't get into. But viruses are something that are as ubiquitous. To clarify, let me back up here just real quick and also just add this here. Viruses are very ubiquitous. They can be found everywhere. This particular type here that you're seeing is called a bacteriophage. We'll talk about that in lecture. They estimate that these outnumber bacteria 10 to 1 on Earth. So these are way more prevalent than even bacteria, which, as we've talked about already, are very, very common. Okay, so these are not rare. I just want to point that out. Viruses are as ubiquitous as any other type of living microbe. Now, another interesting thing about them is that while we always associate them with infection, rightfully so, they cause disease, there's another side to them in that they can actually play a role in the change of an organism's genetic material. A virus can actually infect a host cell and insert its DNA. Let's take a look real quick. This is a figure I pull off the web here from the University of Utah, and it just shows a virus attaching to a cell, inserting its DNA, and then this would actually be an RNA virus, and the DNA then inserts into the host cell here, shown in red. So the virus in this case would actually be an RNA virus. It would insert its DNA, it would actually, or insert RNA, it would convert that to a DNA form, and then the DNA, called the proviral DNA at this point, this is an HIV model here, the DNA form at that point would actually insert into the host DNA. So this is showing the host chromosome representing the host cell's DNA. And what this is showing is the virus RNA coming in, converting to DNA, and then being copied into the host chromosome. So what these viruses can literally do is become a part of the host's DNA. And that's this case is what's called a provirus. We'll talk about that later when a virus inserts into a host cell. But along this line, then, they do this as part of their infection, but there's a consequence to this, and that is occasionally this has a permanent alteration of the genetic makeup. And sometimes these changes can be passed down to future generations, and this essentially constitutes a mutation. We'll talk about this in chapter 9, and we'll define a mutation as any change in the DNA, a permanent change in the DNA. And when that happens, it can be passed down to the next line, and any change to the genetic code is going to have an effect on the organism. Sometimes that effect is bad, sometimes it's good, mostly it's bad. But a mutation is a change in the DNA, and a virus essentially can change the DNA, constituting a mutation. So these viruses not only just causing infection, they're changing the makeup, the genetic makeup of living organisms. And I have an online article for you to read about a theory scientists have that this has actually played a significant role in evolution, and that our DNA is actually a consequence of viral infection. In other words, what makes humans humans partly is infection of a virus, and that doesn't make any sense, but you read about the article coming up in a week or two, and it'll make a little more sense. Okay, let's go ahead and pause it here and just take a quick look at this first section and reflect back on that, and we're going to start the next part here. Okay, so viruses are really, really small. Bacteria are really small, and they are sometimes tens or hundreds of times smaller, even thousands of times smaller. Viruses vary in size, but they're all really tiny. So these are so small, even with the light microscope, which is a basic microscope essentially, we can't see viruses with that. And a light microscope can magnify things about 2,000 times. So they can make things look 2,000 times bigger than what they actually are. And even with that kind of magnification, we still can't see viruses. We can barely see bacteria, but viruses are even smaller than that. Okay, so they require really advanced microscopic techniques or magnification techniques, and that's not something that you'd have access to in a standard laboratory. But we can see them with really advanced microscopes. The microscopes don't let us see them in action. Anytime you see a virus moving, it's always an animation. We don't really get to see moving action pictures, more of a still frame type scenario. That technology probably changing, and maybe I'm incorrect. Maybe there's technology I'm not aware of today that allows that. But as of right now, that's not common at the very least. So these are really tiny. Here's a picture out of the text showing a size comparison. This is a yeast cell. Yeast are a microscopic organism, but they're one of the larger sizes, at least for a single-celled organism. So this is like one of the largest single-celled organisms that you'll find. Fairly large anyway. I shouldn't say the largest, but fairly large. This next to it is a bacteria, an average bacilli, and this is an average cocci. And here we have one of the smallest types of bacteria called a rickettsia bacteria. Okay, so this gives us kind of an average. I probably misspoke a little bit there. There's actually a lot of single-celled eukaryotes much bigger than a yeast, but a yeast is more of an average-sized single-celled eukaryote. And their size varies, is why they're showing two sizes next to each other. They can be bigger or smaller. Okay, but anyway, the point is that these are kind of your traditional average-sized microbes right here. Down over here represent all the different viral sizes. Now, the Mimi virus represents one of the largest ever known. It's actually the second largest. There's another one called the Pandora virus, even bigger. But that's a really, really big virus. Okay, in comparison to the other viruses, this is like a giant virus. And if we look at the size comparison, it's along the same line as the smallest bacteria. And to be fair, this one's getting close to the size of an average bacteria, somewhere in between there. But that's an exception to the rule. Now, we look down here and we see some other size viruses, and these can be similar in size, but we see that they get smaller and smaller and smaller. So they're typically always smaller than your average bacteria, and they can get really tiny all the way down here to a yellow fever virus, which is about a 100th or more, or less the size of a bacteria. You can maybe fit 1,000 of these, for example, in one bacterial cell. Okay, and some of these can get even smaller. And this down here is a molecule, or what's called a macromolecule. So this would be like a really large molecule, and then this is a virus in relation to the average or large size molecule. Next, I want to talk a little bit about the way viruses form and the way that their structures begin, and then we'll talk a little bit about the more detailed aspects of structure. Viruses, as we've mentioned, are more complex than molecules but less complex than cells. And they're more complex than molecules in the sense that they consist of large repeating units of macromolecules that can self-assemble into really advanced forms. So here's a picture I pulled up off the web from the University of Massachusetts from their physics site. And what they're showing is the structure of a virus. And here we see, this is just an example of it's called an icosahedral virus. But you've got some basic building blocks here shown in the top left. And inside here, we have what I believe is showing the DNA or the RNA. I could be wrong about that. I believe that's what they're showing there. But the idea is that they show some of the basic building blocks here, and then they show how these kind of start to form and assemble together and ultimately slowly form a fully enclosed capsule-type device. So when a virus begins its assembly, it starts as these basic components. And what's interesting is that these components fit together in a predictable pattern. And the way they're created allows for this autonomous self-assembly. So these don't have a brain. They don't have a mind. They're just molecules. And yet what makes them so interesting is that they have an autonomous assembly mechanism. So when these particular components are produced, they spontaneously form, and that formation leads to this increased complexity closing in on itself, ultimately creating this entire encapsulated structure, which, as we see here, is the virus. So this is the virus, and this is a step-by-step breakdown or step-by-step assembly of how that virus would form. Now, what's interesting again is when they take over the host cell ends up producing this. So the infection of the virus ultimately triggers the host cell to produce viral components. And then those viral components self-assemble to make more virus. So in a lot of ways, what you really got with the virus is a set of molecules directing a host cell to make more molecules. And we call those molecules viruses. This is kind of difficult to wrap your head around philosophically, but that's ultimately what we're looking at here. Now, a virus is, I'll repeat myself, more than molecules, less than cells. So they're more than molecules in the sense that the molecules are highly organized, and there's a couple of different types of ways that they organize. One is called a capsid. And all viruses, I shouldn't say all viruses, most viruses will have a capsid, which is a protein shell that surrounds the core of the virus, which concludes the DNA, or in some cases we have RNA. In some viruses, not most, they, in addition to these, will package up a couple of proteins or enzymes inside the cell, or inside the virus, They steal this from the host cell along with some of the other things. So you've got, in terms of the molecules here, you've got proteins forming the shell, and then you've got nucleic acids forming the core, which consists of DNA or RNA. And then occasionally they'll package up a few enzymes in there at most. And if we look at it here, this is just a layering with the covering on the outside and the core on the inside, obviously. And just to reiterate this sentence up here, this is a very minimal set of molecules. It's more than just basic molecules, but this is essentially like, think of it like a little kit of what you need to break into a host cell. You've got protein and the nucleic acids and sometimes a few extra little tricks, and that's about it. And viruses essentially are minimalists in that they only consist of what they need to infect. So they don't, to clarify, they don't have a life outside the host cell. Nothing happens until they have a host to infect. That's their entire quote-unquote existence. So looking at this next slide, we see some of the structures. We have a number of different types of viruses, as you might imagine, but we can divide most of these into one of two generic forms, and that covers the majority of them, at least from a fundamental standpoint. And with one type, we have what's called a naked virus, And with another type, we have what's called an envelope virus. And if we look here, the naked virus consists of, this is actually what's called an icosahedron pattern. But this kind of odd shaped pattern here represents the basics of a virus. This is actually the core, this is actually the capsid or the coat, and then here inside we have the core with the DNA, the RNA. If we look at an envelope virus, we essentially have this same thing inside here, this same icosahedron shape as we see here, but now we have an extra layer around it, and that extra layer is called the envelope. So we call these envelope viruses. But to point it out here, we have basically we have a basic virus here, we call it a naked virus, and then we have a more advanced form which consists of that plus an envelope. Okay, but they're very similar. That's why I want to point out the difference here. We have really kind of the same thing with one more advanced than the other. A couple things that are important to point out here. This light blue structure with the weird shape is called the capsid. They don't all look like this, but that's a common shape. We see then the nucleic acids inside, and then another key part of the capsid is these little things that stick out here called spikes, sometimes called peplomers. And a spike is essentially what the virus is going to use to grab onto a host cell and attach. Over here then, we see a similar type of structure, except instead of the same type of spike, we see a variation of that, and these are embedded within this envelope here. But essentially they're called the same thing and embedded, in this case, within the envelope as opposed to the capsid. But these also would be used to grab on or attach to the host cell. So the capsid essentially represents a protective layer for the DNA or the RNA inside the virus. And ultimately you can look at the DNA, the RNA as the kind of critical aspect of what the virus is. So really the virus is what its DNA consists of, and if that makes any sense. The point I'm trying to get at is that the virus really consists of the DNA, but the capsid is there just to protect that. So you can almost think of the virus's DNA or RNA as the brain, or maybe the heart, or both. And then you can think of the capsid as kind of like the body. So the body is nothing without the brain, and vice versa. So they need each other. But the capsid is more of there for protection, as well as for movement, and as well as for transportation of the DNA from place to place. And this is ultimately what's going to spontaneously assemble from those components as I showed earlier. Now, the way that the capsid is able to form, as I mentioned, it's made up of repeating subunits. And these repeating subunits have a symmetry that allows them to spontaneously form together. These repeating subunits are more specifically called capsomers. So if we go back to that figure here real quick, each one of these would be considered a capsomer. And this entire structure here would be a capsid. And those terms get confusing real quickly. But capsomer, individual, capsid, entire. You just have to commit those to memory. Most of the capsomers form one of two shapes, one of which we already showed. And just to back up here, do I have another picture? Yeah, here's another picture. I'll go right back, sorry. This is an icosahedron here, that's one. That's what I showed here. This is a icosahedron. The other common type is called a helical, and you can see a real image over here. This is a kind of a illustration, obviously, but cut into a cross section, showing it half down the middle. And this is what it's showing, a helical capsid being formed spontaneously. So these capsids represent the whole structure covering the DNA up. Each one of these little individual pill-looking structures here represents the building block of that capsid, or the capsomer. Okay, these are going to be different with each type of virus, and especially with the type of shape it has. So if it's a helical virus, it'll have a certain type of capsomer. Icosahedral viruses will have another type of capsomer, and then they'll vary even within those. Okay, now the capsid directly covers the DNA. If we look at an envelope virus, such as this one here, I believe this is showing the HIV virus, or maybe it's the influenza virus, one of those two. We see that the capsid is tucked away inside of the envelope. So this is a more advanced virus. It's got, in this case, many capsids tucked away inside a larger envelope here. So this would then be, the envelope would be another protective layer for the capsid below, which is even also protective to the DNA. So these really advanced viruses can be multi-layered. Simpler viruses have just the capsid. Okay, so helical rod shaped, they form bracelet-like discs or barrel-shaped capsids, and then a icosahedron, which is defined as having 20 sides and 12 evenly spaced corners. And that's one of the probably hundreds of weird random geometrical shapes that you'll never hear again in your entire life. So you just have to remember that one for this class. Okay. There are a lot of exceptions to the common rules of shapes, and one exception is the idea that we have what are called complex capsids. So these are another category that kind of you throw shapes into if they don't fit real easily. So for example, you have what's called a bacteriophage. We'll get into more detail on these later. But if you, as you can see here, this whole structure is the capsid. Illustration here, this is a real image here. These really exist. That is an actual picture of them. Looks like a little spaceship, but that's a virus. It's a bacterial virus. These types of viruses have a very complex capsid. So this whole thing is considered the capsid. So this, rather than having the name of a icosahedron, which it has that basic shape, it has more to it. So we call this a complex capsid. And there's other types of complex capsids. If we go back just real quick and we look at the picture I showed you, we see some complex capsids on here. So for example, here with the Mimi virus, this would be an example of a complex capsid. So there's other ones out there, but these are highly variable. So we're looking here at just the basic types of capsids, and this is an example of a complex one. Okay, so that's some basics of the structure. Hopefully that makes sense. Go ahead and stop here and take a look at part 2. So next, I want to talk about the envelope viruses. Envelope viruses have this extra layer that we mentioned already. It's called the envelope. And this is essentially just an extra layer of protection, fundamentally speaking. Anyway, it helps ensure that the heart or the brain, as I analogized earlier, the DNA is protected in order to get to another host cell. So the envelope, in a fundamental sense, kind of provides that extra layer of protection. But in addition to that, it affects the way the virus actually infects a host. So it changes the way the virus interacts with the host. And in fact, the envelope actually comes from the host cell during infection. There's kind of a chicken and an egg paradox here. But as we see it today, the envelope of a virus actually is taken from a host cell during infection. And when the virus is exiting the host cell, it literally coats itself with part of the membrane from the host cell and then takes that with it as it exits. It's almost like the virus is wearing part of the host skin as it exits the cell. Kind of creepy. These types of viruses are unique to the animal kingdom and not known to parasitize other groups of organisms, at least as far as I know. In 2016, this information changes, but fundamentally speaking, these are unique to the animal kingdom. So we don't see this in, say, plant viruses or bacterial viruses. So as I mentioned, the envelope essentially is acquired from the host cell. They actually take that during infection. I've got a quick figure of that, and I'll go right back. As the virus is exiting the cell, it layers itself into the membrane by first putting down some viral proteins. It then attaches to those proteins and then very slowly pushes itself outward and coats itself in the host cell host cell membrane as it pinches away from the host itself. So as it exits, it uses some of its own protein along with some of the host cell membranes to coat itself, and then takes that with it and exits on and goes on to the next cell. So this is an illustration here. This is actually a real microscope image showing step by step the process of which a virus is actually exiting from a host cell. So that's a real picture that's kind of cool. Artificially colored, but that's actually a real thing there. So This is kind of interesting. But the idea is that the virus takes from the cell what it needs to go on to infect another cell. This has consequences, and when we get into the damage in the lecture next time, one of the things I'll point out is that a host cell can only lose so much membrane before it becomes permanently destroyed. So one virus taking a little bit of the membrane won't be too devastating, but typically a virus exits in the order of magnitudes of, say, millions. So one viral infection can lead to millions of copies, meaning millions of little pieces of the membrane are taken, and that just depletes the membrane and causes it to disrupt. Okay? So ultimately, the envelope virus is unique to animals and is unique in the sense that it steals from the host cell during that infection. The last part here I want to mention is, before the next part, I want to mention the nucleic acids, the core of a virus. All living things consist of both DNA and RNA. This is one of the more technical aspects when we define a living thing. They have DNA and RNA. Viruses have the same types of material, but will typically have either DNA or RNA. But interestingly, they use them in a very similar kind of way. So viruses have the same building blocks of life that living things have, which is DNA and RNA. But they use either one or the other, and they use it in a way that's similar but different from living things. So there's a lot of fundamental things that are similar between viruses and living things, but they're different enough that they don't really match up perfectly. But the fundamental comparisons are there, and that the heart and soul of a living cell is DNA. That in all living things, DNA is what guides the activity of a cell. The same is true for viruses. The exception is that sometimes they use RNA instead of DNA, but that still is true that they use one of these two to essentially guide what it is they're doing and direct the activities of the virus. So fundamentally speaking, those are very similar properties of life and properties of viruses. It's everything else around them that doesn't match up. But ultimately, that's what we see here. And let's just pull up an image real quick of DNA and RNA to make sure we know what we're talking about here. Okay, so DNA and RNA are really complex forms of molecules that have genetic information, kind of like a blueprint. This would be a computer animation, but an accurate anatomical model of a DNA molecule. I'm assuming that's an animation. It's hard to tell these days. Here we have more of just an illustration showing a DNA molecule compared to an RNA molecule. So these consist of basic molecule building blocks, and they're arranged either into a double helical pattern or a single helical pattern like this. Similar but different. But these contain the blueprints of life, and this is the case for both viruses and living things. So the question down here then is, what is the central dogma of biology? And the idea behind the central dogma of biology is that, you just want to jot this down here. The idea is that DNA directs the activity of RNA, and RNA directs the activity or the production of protein. So fundamentally speaking, this is the central dogma of biology. What this really means, though, and what the central dogma is ultimately stating, if you want to type this out, this is a figure diagram expression to represent what we're talking about. If we want to actually try and form a coherent sentence to describe this, we would essentially say that the central dogma is that, how about I get it in a font you can actually see? There we go. In all life forms, really? Sorry, guys. In all life forms, DNA directs the production of RNA, and RNA is directly translated into protein. That's not the textbook definition, but that gives us an idea of what I'm talking about here. So this is a fundamental property of life in that DNA is used to make RNA, which is then used to make protein. Okay. Now, what I like to do is add just a little bit more to this because I think it really helps you go one step further. And I'll draw this in red. This is not part of the central dogma. This is part of my interpretation to help you understand it. And to kind of summarize this here, protein, ultimately, why that's important is because protein directs the activities of the cell. And I usually say directivities / work of a cell. So ultimately, protein is what makes a cell tick, if you want to think of it that way. Protein makes a cell tick. Okay? I'm not sure if the K goes there, but I think the K does go there. The idea is that proteins are what make cells function. You can think of these as like the building blocks of a cell, the most important types. Another way to think of proteins, you can think of these as the quote-unquote workers of the cell. So they do the work, and in a lot of ways, proteins are the quote-unquote workers of a cell. Okay, so this is ultimately what gets stuff done in a cell. It's like producing a machine that allows you to build things. DNA essentially has the blueprint that allows the cell to make the machines that allow the work in a cell to get done. Okay, and the central dogma simply states that all life operates under this basic set of principles, that DNA is used for this purpose. There's a conversion process here. Protein is ultimately then used in this sort of manner. That is the central dogma of biology. Finally here, let's talk about how this relates to viruses. So this whole process here, we'll package this up. This whole process relates to the way a living thing operates. A living thing directs all of this activity simultaneously, DNA into RNA into protein. A virus, however, what it's going to do is it's going to come along and it's going to insert itself somewhere into this process. And instead of churning out the workers for the cell, we'll clarify that the proteins here are cell workers. They work for the host cell. Clarify more, these are host cell workers. So they function for the cell, as you obvious, right? But when a virus comes along, it actually redirects the cell to make proteins that are now no longer for the host cell, but are instead proteins for the virus. So the virus is going to make viral proteins. And it's going to do that by using its viral DNA, for example. So we have a DNA virus. And the DNA virus is actually going to insert itself into this process right here. So its DNA looks the same as the host DNA. So it uses that same type of DNA and uses it to trick the host cell into making copies of the DNA into an RNA form. And then ultimately the DNA virus creates viral proteins through the host cell. Think of it as like taking over a factory that makes, insert any example here. It makes widget A, whatever that is. And instead of making widget A, the factory is hijacked and now makes widget B. Okay, so it uses the same machinery, but redirects the products to make something different. And in this case, it's making viral proteins. And ultimately, these viral proteins, they are going to consist of capsomers. They're going to also consist of building blocks for DNA and RNA. So they're going to be used to make viral DNA / RNA. So these viral proteins are ultimately going to make more viruses. Essentially, that's what's going to happen here. We're going to ultimately get more virus from the viral proteins. So they essentially are the viral workers, and they're going to make more virus. So the central dogma gives us an idea of how life works, and then it gives us an idea of how viruses fit into that framework. And they kind of, from the outside, squeeze in and hijack the framework of life and the process of life to fit themselves into it to make more copies of themselves. So you kind of get an idea how they're similar to life, but also fundamentally different. And scientists have just decided that they're not life, and have decided that life is this and viruses are not that. So they are considered these infectious particles. But I like to point out the fact that they're really quite more than that, and they have life-like properties that exist in kind of a weird way that's dependent on other life. So they're not independent, but they are very much alive in contact with other host cells. Okay, so that's the central dogma of biology and how viruses relate to it. All right, so here's part 3. Go ahead and stop, and we'll move on to the next part. Okay, next we're going to get into viral replication, and we're going to take a look at some of the fundamental steps that relate to how viruses actually infect a cell and what's happening there. As this slide is talking about, there's lots of complexity here. We're going to focus on animal viruses, the most relevant type to us. We're going to focus on those, and we're going to focus on the overall process as opposed to lots and lots of detail in these specific steps. And we're going to focus on the fundamentals of how these applies to most viruses. Okay, so one of the first things you want to recognize, and what you'll be definitely tested on later, are what are the 6 steps of the virus cycle. This is called the virus life cycle. You can call it the replication cycle. Steps or phases. Okay, so number 1, that's actually not a typo. It's actually adsorption, not ab, but adsorption. Penetration, encoding, synthesis, assembly, and release. Today, we're going to talk mostly about this first step here. And this is what I think is fundamentally the most important and the most applicable to understand. In other words, this tells us a lot about how viruses function in addition to just describing the life cycle itself. So it gives us some idea of how they function. So we're going to focus on that part today, and then you'll look at the other steps later. So the first step, adsorption, means to adhere to a surface. This term could be used in other contexts. Many different molecules absorb to the cell membrane, for example. But in this case, we're using it to describe how a virus, shown here, adsorbs to the host cell membrane, where it's going to start the infection. Okay? It's important to point out that this is always, caps, required before a virus can invade a host cell. At least in animal cells, it is. So adsorption, attachment is always required. So why this is important is because the way they attach determines whether a virus can even cause infection in the first place. If they don't attach to the host cell, they can't cause infection. So to clarify, you could have any virus. Let's take the worst case, one of the worst case examples, and say the Ebola virus. You could have the Ebola virus come in direct contact with tissue, but if it can't attach to that tissue, it can't cause infection. Now, unfortunately, the Ebola virus can attach, but the point is that any virus, no matter how potentially deadly, can't do any damage unless it actually physically latches on to the host cells, and that's the first step during the infection process. If that attachment or adsorption never occurs, then none of the other steps that occur after that, listed here, none of those other steps will take place either. So this is an absolute prerequisite to infection, and without this step, none of the other steps proceed. So let's add to that then. When the virus is adsorbing, what it's specifically attaching to are the host cell's receptors. Receptors occur on all cells, and they essentially are used for purposes such as entry in and out of the cell. Sometimes they're used to attach cells to other structures, but they fundamentally have some reason for being there that has nothing to do with the virus. So all cells have receptors for a variety of purposes, and these receptors are built into all cells as part of the fundamental functions of the cells. What viruses do is they hijack these receptors and use them as a way to get into the cell in a way that the cell never intended. Think of it like a door. A receptor is like a door, and we put doors on every building because we want to get in and out of them. And if you put a door on a office building, or let's say a store of some kind, a place where they're selling something, you put a door on it because you want to allow people to come in during the day, but then you lock that door at night because you don't want people coming in after hours, right? A receptor is kind of like that. It's used in one capacity, but can be exploited in the sense where a door can be busted open at night and somebody can get in the door that way. So you have a purpose for it, but then a way to exploit it in a way that wasn't intended, such as breaking into a door when it's supposed to be locked. Receptor is kind of like that. It's got one purpose. The virus uses it in a different way. But ultimately, that's how the virus gets into the cell. It attaches to the receptor, hijacks those, and gets in. These receptors were called glycoproteins. This is a sugar protein molecule. So it's part protein, part sugar. And this is a typical type of molecule that the receptors are made of. And just recognize it's an entry point used for the cell for one reason, hijacked by the virus and exploited for entry. Okay, so that's a real quick part about adsorption. And then we'll talk about the next section here, which is how this relates to the way a virus infects certain types of organisms. Okay, and then that'll be the last part. So this will hopefully be a short session today. So the last part is adsorption and host range. And the idea is that in order for a virus to actually attach to the host cell, there has to be a certain compatibility or match between the virus and the host. And the virus has to have a certain ability to attach to the receptor of a host. And this ability is something that only occurs between certain viruses and certain hosts. This is what we refer to in biology as lock and key specificity, meaning that like a lock and a key, they have to have a certain amount of ability to interact that allows them to be compatible. In this analogy, let's type this out here. In this analogy, the virus is like the key, and the host receptor is the lock. So the host cell has a lock, and the virus is essentially going to come along and enter by attaching to that lock and opening up the door, as we analogized earlier. The ability to do this has to do with an interaction between the spikes of a virus and the host cell receptor. So if we look back here real quickly at this slide, we get another visual reference here to the spikes. These occur around the capsid of a naked virus and then occur in the envelope of an envelope virus. And then if we look back at this figure here real quick, a simpler figure, but it shows these red dots as these are the spikes, and this is the host cell it's attaching to, and these are the receptors here. Okay, so those have to fit together like a lock and a key. If that happens, then the virus can successfully adsorb or attach. Now, the truth is this doesn't always happen, and in fact, in most cases it doesn't. So let's do another quick little diagram and just show a couple different scenarios here that could take place with a virus. Okay, here we see a diagram I've set up showing 3 potential host cells and a virus here with a particular shaped spike. So this here is the spike of the virus, and I've drawn just a generic shape here. Now, to point out something here, these are not drawn to scale. So this whole thing here represents a virus. I know it looks nothing like a virus that I've shown you so far, but this is simply for demonstration purposes, not to be structurally accurate. So this virus here, this is the body of the virus, or the capsid of the virus. This is the spike drawn in extreme exaggeration. And over here we have host cells that would be much bigger in reality. Okay, but the point is we have 3 potential host cells, each with a different shaped receptor, so to represent a different lock-like configuration. And then over here we have a potential virus that's got a very particular looking spike. Okay, now as we might already start to imagine, this virus is only going to be able to infect one of these 3 host cells here. And you might already be able to guess which one it is, but let's start with this one down here. If we look at how these two would potentially match up here, we can see that this particular shape is not at all conducive to fitting into that lock, okay? Same thing here, right? It's not going to fit. The shape of the spike and the shape of the receptor are not compatible. And lo and behold, this last one here, these would theoretically fit together, and the virus would be able to attach to that host cell and absorb rather effectively. Okay, so ultimately, this shape of a spike in this particular type of virus would dictate that it would infect host cell A and not B or C. Okay, now as we'll talk about a little bit further here, the host cells can be different species, or they can be different cells within a single species. So for example, this could be a liver cell. There are viruses that only infect the liver, such as the hepatitis viruses. This could be, say, a skin cell, and this could be, let's just say, a muscle cell. That's another random example. So these can be the same organism and actually different cell types. And in some cases, a virus only infects, say, the liver because it only is able to attach to liver cells. Some viruses can infect multiple cells. Some viruses can infect only one type of cell. So there's a lot of different compatibility there. Okay, but that's just a quick visual for how the spikes fit in with the host receptors and how that would dictate adsorption. Now, this last part here is what's significant about this. This dictates what we refer to as the host range. So the ability of the virus to absorb and fit in with the lock determines what types of organisms and what cell types it can actually infect. Some viruses can infect a wide range of host cells because their spikes have lots of different compatibility, lots of different compatibilities with multiple locks or receptors. You can think of viruses with a broad host range as viruses with like a master type key. They can open a wide range of doors with the one key. That's somewhat simplifying the whole scenario, but that's essentially what's happening there. They can get into many different cell types with the single type of lock. Single type of key, excuse me. So broad host range is one example. Another example is that some viruses have what's called an intermediate range. They can infect a couple cell types, but not a broad range. So just kind of a lesser extent. And then we have the third example, which is restricted. And in this case, they can usually only infect a very specific type of cell, one cell type or one very, very specific species or one cell type within a species. Go through a few examples here. The first one is an example of a virus with a broad host range. Now, this is actually not that common. Broad host ranges, at least from a traditional standpoint, are not the norm. But one example is the rabies virus. The rabies virus is something that we're all familiar with, and we know that we can get it from animals. Animals get mean and aggressive when they get infected. That is somewhat true. Over-exaggerated, but still somewhat true. I think that's the old Yeller picture there. But what happens here is that the virus infects brain tissue, and more specifically, it's infecting a type of receptor in the brain called an acetylcholine receptor. Now, these receptors can actually occur throughout the body, not just in the brain. But ultimately, the effect carries over into the brain. So the acetylcholine receptor is a receptor that has actually been conserved throughout evolution and is found in most warm-blooded mammals. So while you and I and a raccoon and a dog may all be very different, from a basic fundamental standpoint, we have identical brain receptors called acetylcholine receptors. These receptors haven't changed that much in the course of evolution, and all animals share virtually the same receptor. So for this reason, the virus can infect all different types of animals because what it's attaching to is found in all these different types of animals. So because we all share similar chemistry with our brain receptors, the virus can infect all of us, and that's why it has a broad host range. So in this particular case, it has to do with the fact that its attachment point is found in a wide variety of animals, mammals specifically. So that's for an example of why we see a broad host range in rabies. On the other side of things, looking at a restricted virus, a restricted virus would be an example, or an example of one would be the HIV virus. HIV it stands for the human immunodeficiency virus. This is one we're probably familiar with, and it infects people, as we know, and is sexually transmitted. But to be more specific, HIV actually only infects one cell type in the body, and it infects a cell type called a CD4 T lymphocyte, which is also known as a type of helper T cell. We'll talk about that later. You don't have to write that down. I'm just putting that there for you. The CD4 T lymphocyte, also known as a helper T cell. These cells are critical to triggering a full immune response when you're infected by a virus or infected by some other sort of organism. They are essentially the alarm signal for the immune system to kick into full gear. And they are absolutely critical cells. When HIV infects the body, it targets these cells very specifically. And when it does that, it actually starts to damage the body's entire immune response system. And this is why ultimately, if HIV is not treated, it leads to full-blown AIDS, the acquired immunodeficiency syndrome, in which your body becomes susceptible to a wide variety of infections. You're probably familiar with the fact that if you have HIV and AIDS, your immune system is depleted, and you're likely to be infected and die from a variety of different infections. I'm not going to get into that whole topic here, but I'm using this as an example to discuss how it works and why it's considered restricted. So to clarify, it's restricted because it only affects one cell type. And to add to that here, because that one cell type is critical to the entire functionality of your immune system, by infecting that one cell type, the virus has the potential to just devastate the whole immune system. And this happens slowly. HIV infection usually takes at least 2 to 3 years to develop into full-blown AIDS, and sometimes it can take longer than that, up to 10 years in some cases. So this is a long, drawn-out process. Doesn't happen overnight, but slowly over a series of months and years, the T cell count, the helper T cell count, gets lower and lower and lower as the infection progresses higher and higher and higher, and eventually it just completely wipes out the signaling response by your immune system and leaves the host completely compromised. So this has to do with the fact that the one cell type it infects is the one cell type absolutely critical to your immune system, and that's why HIV is so devastating. The truth is, there's lots of infectious viruses that cause infection of our body that we are never even aware of, or that aren't really serious, and that's because not all cells in our body are quite this critical. Most cells are fairly important, but a lot of them are fairly expendable. So in the case of HIV, it turns out that the reason it's so significant, or at least one of the reasons why, is because it attacks this very critical cell. In fact, this cell type right here is actually critical to removing viruses from the body. And one of the things it does is signal the rest of the immune system to come in and remove the viruses from your body. We'll talk about this in more detail later when we get into the immune system. But that kind of explains why HIV is so devastating. Even though it only affects one cell type, that one cell type is critical, and it actually is necessary to remove viruses. So HIV, being a virus, is actually infecting the one cell type that the body needs to remove viruses. And that's why it's so devastating. And then that carries over into other aspects of the immune system. And ultimately, the host is not able to fight off any sort of infection, viruses or otherwise. Okay, so that's a little bit about the early stage of the infection process, adsorption. So, as I said, we'll take a look at this further in class. We'll carry over into the next stages of the life cycle, and then we'll get into some of the damage the virus cause, and follow that up with a slightly different topic on viruses that infect bacteria. All right, hope you have a great day, morning, afternoon, evening, and as always, email me with any questions, and I will see you next week in lecture.